Now we are moving to the reverse chemical genetics. To remind you the difference between fold and chemical genetics, fold and reverse chemical genetics. One was a target to system, and the other one was a system to target, right? Reverse chemical genetics is you define your target first, and then check whether what is the effect into the whole system. So that's why the first one, target to system, should be reverse chemical genetics. This is biochemical pathways known to human being. And this one, in a sense, looks like a quite complicated, but actually this is not representing everything going on in our body. It may be only 1% or even lower, smaller than the whole biochemical pathways in real situations. Anyway, still this is quite looks like a complicated. Now, if you decide your target among these pathways, that should be starting point of reverse chemical genetics. Each small point there, it can be maybe one protein, or you can choose, rather than just one protein, you can choose one signaling pathways, like one cycle like this. So I want to study this protein, or I want to study a long line of this cycle. That can be starting point of your decision of which target you want to attack or study. For example, cell cycle. When the cell gets growth signal, now the cells getting into the so-called cell cycle. Originally, this resting status we call G0. This is gap 0. Now, once getting to the cell cycle, the first step is G1. To make another copy of those cells, first thing is collecting a lot of materials first, DNAs and proteins and all those materials, to secure that whether the cell has enough resources to make the body of those cells. Once all the checking is done, then first checkpoint here, and if this is okay, then it goes into second step of synthesis of DNA. Remember, each cell should have one copy of genetic information, and those are stored in DNA. So most important part of cell division is making exactly same copy of DNA to donate to the daughter cells. So that's why this second step of synthesis of DNA is most important. If this synthesis cannot be completed for whatever the reason, then the cells should not go into the next step of division. But synthesis is perfectly done, then second checkpoint here, okay, synthesis is clear. Then it goes into the second step of preparation, so-called the G2 step. And then still everything is okay. Now it goes into the real cell division, so-called mitosis, that can happen. During those phases of a cell cycle, there are a lot of checkpoint the proteins are involved here. Most of those proteins are kinases. Cyclin-dependent kinase 1, 2, and 4, 6. Starting from the G0 going into the G1, in the beginning, the CDK4, CDK6 is involved, and CDK2 is the first checkpoint here. And then in the middle of DNA synthesis, another partner, cyclin A dependent CDK2, and then eventually in G2, CDK1. So multiple numbers of kinases are acting as checkpoint the proteins. So this is one example of, in the beginning, how you decide your target. This CDKase has been very important target for cancer therapy because these proteins are the checkpoint proteins which can decide whether the cells can divide or not. Then, for example, if you attack this kinase, then kinase does not give you the go sign, the cells cannot divide, cancer cell cannot divide. So this is one possible way how you decide your target protein in the beginning. Here are some topics for reverse chemical genetics. And unfortunately, I could not find the Wikipedia reference for covering all of this. So instead, I put up the one review paper you can look up. And this paper, you can easily download from a library of NUS. So this much, I will not provide the PDF file of this. You try yourself to download this the reference. This is part of the training, how you collect your necessary information. In cell cycle, the examples I gave you, some kinases can be potentially good target for a small molecule. Then let's see what is the function of those kinases. Kinase is an enzyme, and then they utilize ATP as a cofactor, and then transfer one of this phosphate to the substrate. The substrate can be protein, and if it is a protein, it can be tyrosine, threonine, or serine of those hydroxyl group can be phosphorylated by this kinase. And opposite direction of this dephosphorylation is catalyzed by phosphatases. But not only proteins, many of those other small molecules like a carbohydrate or lipid molecule also can be phosphorylated by kinases. So anyway, kinase can be important target for small molecule or drug development. Again, this for kinase the action. Now, this is a serine substrate, this is a tyrosine substrate utilizing ATP. ATP is donating phosphate to the target, and then it is hydrolyzed here and generating ADP as byproduct. Then there are several different points how you can monitor this reaction. Maybe you can monitor the changes of a product, or measuring those amount of ATP, or generated ADP amount or phosphate amount, kind of a different the screening format that you can set up. And if possible, you want to make this into color changing the method, then it's very easy to automate those measurements. So this kind of the test tube test of a biological the function, we call this test as in vitro test. In vitro means test tube. So inside the test tube is an in vitro test. But let's say if you have hundreds of thousands of small molecules and you need to test them very quickly, then one by one, using this kind of a test tube or one qubit, it can be quite time-taking and also highly the labor-intensive approach. You may want to have much faster approaches. So that kind of fast the screening method we call HTS or high throughput screening. At least utilizing this kind of plate instead of a one by one those qubit. This plate usually 8 by 12, so this is 96 well. And using this kind of a multi-pipette, you can add at least 10 or 12 the reagent at the same time. Then at least from one experiment, you can measure almost 100 tests. So again, to automate this system, it's better to have every system is somehow color introducing, color generation, or color changing. Now, next topic is orthogonal chemical genetics. Orthogonal means it's crossing at right angle, 90-degree angles. So this is a mathematical other terms. They overlap, but they don't affect to each other, kind of meaning it has. So let's see what it means. I said kinase is involved in cell cycle, but kinase is not only in cell cycle, and many other cell activation pathways. Almost every point, kinases are doing very important roles. Here we can see a lot of kinases. RTK, you don't need to know the exact name for all of these. Receptor tyrosine kinases. Anyway, this is kinase. And then another kinase here, and kinase, 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 kinase. So those kinases has multiple the signaling pathways, and more complicated the situation is they crosstalk. Not only those the single line going down, sometimes they crosstalk, or sometimes even they do feedback going up. So it's kind of a very complicated network they form. In human bodies, we believe about 500 kinases exist, and those network is quite complicated. So this is called as kinase network, and whole kinase set we call genome. Now, we have 500 different kinases, but one common factor is most of those kinases, they use ATP as cofactor. That means all the kinases, they may have one ATP binding site and maybe another substrate binding site, or there is another partner coming out and binding here. So one common factor is ATP binding site. Even though we have 500 different kinases, all of them has a very similar binding pocket for ATP. That's something we can assume. That means if your drug development is targeting this ATP binding site, actually it's quite difficult to differentiate different kinases because the binding site has almost similar structures to each other. Also, another problem is, let's say, if you want to know the substrate of this kinase, how can you do it? Inside the cell, there are many kinases exist together. If you add ATP and monitor which protein is phosphorylated, then this ATP can be utilized by all other kinases at the same time. Then, actually it's very difficult to figure out what is the direct substrate of this kinase. To solve the problem, here is one other trick. So let's say this is your target kinase, and if you add ATP, then your substrate can be phosphorylated like this. Here, this ATP binding site you can modify. So kinase is protein. So this should be composed of amino acid. So originally it has bulky amino acid group here. Then you change that with a smaller amino acid. So that's why you make some kind of small hole here. And because of this hole, original ATP cannot bind here tightly. So that's why this modified kinase cannot do the proper catalytic function as a kinase. Now let's think. How about we make this ATP is a slightly the different structure with some kind of small bump which can match here. Then this kinase, this ATP, modified ATP, cannot bind to original kinase because of this other bump. But if this modified one meet with this changed ATP, it can be perfectly matching and it can make substrate, it can make the catalytic reaction happen. Now it's possible that, let's say, all other kinases, normal kinases, they utilize regular ATP, and this changed ATP, if you can introduce radioactive gamma phosphate, then only the substrate of this protein, this kinase, can be phosphorylated with radioactive phosphate. How about it? So for example, let's just say in kinome, inside the cells, there are hundreds or 500s of different kinases are there, and only you choose one kinase, in this case, siRNA the kinase, and then this one you introduce this kind of a hole, and then you make ATP, which has this kind of a bump, then only siRNA can take up this ATP, and then all other enzymes, all other natural kinases, they utilize regular ATP, and if you run the gel, all other proteins is phosphorylated here, but only radioactively labeled one, it is phosphorylated by this modified the siRNA kinases. Sounds like a very crazy idea. This is like competing with billions of years of evolution that optimized the binding of ATP to the kinase. Using the same technique, now it's possible that you can modify one by one of different kinases, and then you can make those big map among those very complicated kinase network. Which protein is phosphorylated by which kinase, kind of mapping is possible. This challenging project has been proposed and performed by a scientist named Kevin Shockett. He proposed this ambitious project from his very early stage of career, and he's making the progress in this field. Exactly the same idea of a bump and hole strategy can be applied to inhibitors. As I said, if you attack ATP binding site for the drug targeting the site, selectivity is quite difficult to achieve. So in this case, among many different kinases, you select your target kinase and then make this a whole, and then you make this bumped the selective inhibitor, then among complicated many different other kinase other network, you can selectively inhibit only one kinase and then check the result. Then you can figure out what was the function of these kinases from this very complicated network. Quite amazing technique, isn't it? Now let's move next topic. Disruption of protein-protein interactions. When we draw one protein, usually we have one line of a primary structure and then folded into secondary or tertiary structures. But actually we look at the inside of those proteins. Each protein has its modular structure. Some of them is a binding site and some of them is a reaction site. So one protein can be analyzed by different modules. Same thing can be applied to different type of proteins. Now by looking at those protein modules, so let's say tyrosine phosphate site is binding site for the SH2, so then we can think of those connections, which protein may have a binding to which protein, kind of a network we can think. Our genome number is surprisingly low, about 20,000, and then even though we have a bunch of post-translational modification, and we have a different structure of proteins, but those proteins, how they interact with each different protein is the very important the fact to understand those functions and also network of those protein sets. To study those protein-protein interactions, here is one very common useful technique. This technique is based on transcription. Transcription is a new mRNA synthesis, right? So to start transcription, you need one motif, DNA binding domain, so-called DVD, which may bind to the promoter area. And then activation domain, so this can actually start those gene expression. This activation domain may include some mRNA, the RNA polymerase kind of things. Utilizing this machinery, we can modify this structure like this. First of all, in DVD, we can introduce one protein, protein A, and AD, another protein, protein B. Only when these two proteins bind to each other. If they bind to each other, DVD and AD is close enough to each other, then gene expression can start. So, if gene expression can make color generation or fluorescence generation, now this is your readout to see whether this protein A and protein B is binding to each other or not. Do you follow me? So utilizing this system, now you have two partners. Two partners are hybrid together, then you can read out. This system is called as a two hybrid system. Yeast has been used as a popular model for this. So sometimes this technique has been used, yeast two hybrid system. The goal is to check whether these two proteins, protein A and protein B, whether they bind to each other or not. And readout is utilizing this gene expression. Clear? Then using this system, let's say if you want to know what is the protein partners, binding partners of protein A, then you can fix this DVD with the protein A as one system. And then you can make a library of these different proteins, let's say from the cDNA library. cDNA library is from one cell, you can collect all those mRNAs, right? And then those mRNA can be attached to this AD structure. Then you have a library. Each cell has only one component of this library protein. And when they bind to each other, only those matching one, it can generate this gene expression. This is a two hybrid systems. As an end result, you can get the information that the list of binding protein for protein A. This is interaction database. That's why this kind of data set we call interactome. So genome, transcriptome, proteome. Now we are talking about interaction of two different proteins. Only when they interact together, they have a function. Then this interactome is very important information. Okay, let's say to inhibit the kinase, there are two different kinds of approach. One is this kinase has a partner and it is binding to another protein, and this complex is the active kinase. One approach here is to add ATP binding site inhibitor. If you add this inhibitor, replace this ATP, and then this protein complex cannot do the catalytic functions. This is one way. The other way is, rather than this ATP binding site, you target this site and add this inhibitor, then the partner protein cannot come and bind here. Then this is the indirect way, not directly attacking those functional part, but by interfering these protein-protein interactions, then still we can get same result of inhibition of this specific kinase. This is a targeting approach using disruption of protein-protein interactions. Next example is chemical inducers of dimerizations. If these two protein motifs, when they get together, if they can get activated, one way is you can add this small molecule which can dimerize these two. Then they bind in between here and they recruit this protein to the membrane and activate it. In a sense, this is quite similar to two hybrid system, right? And in this case, this small molecule, this dimerizer, this can be activator of this protein by inducing those dimerization or inducing those protein-protein interactions. After this is activated, now you can add another competitor. Let's say in this case, it has two binding sites. One bind to the D1, one bind to D2. Now you add another competitor which may bind to the only D1. Then it can induce dissociation of this. In a sense, this is opposite way of dimerization, which is similar to protein-protein interaction disruptors. In this case, if you don't add a dimerize, add only competitor here, then you block those two proteins, the interaction to each other. Next approach is a targeted protein degradations. The approaches we have seen so far is utilizing chemical tools. We attack those active site or protein-protein interaction disruption or inducer those dimerization kind of approach. In this case, we target the actual physical degradation of proteins. Let's see how we do. In our body, we have a protein named as a ubiquitin. Sounds familiar to you? So this was one of the post-translational modifications, ubiquitination, right? And this ubiquitin is death mark. Molecular weight is approximately 8.5 kilodalton. And utilizing the big machinery here, this ubiquitin can be added to any target protein. Once ubiquitin is added to target protein, now this target protein is kind of sentenced to death. And then, recognizing this ubiquitin, it goes into this big complex so-called proteasome, and the proteasome just break out the protein. Proteasome does not care what is the structure of this protein. It just only recognizes only ubiquitin, the polymer, and then ubiquitin can be recycled for another the death mark, and then this protein is degraded into smaller pieces to recycle for amino acid other utility. And this proteasome is one of those biggest machinery in our body, has a shape of this kind of a tube, and then the protein can get inside here and then just ground up and just break out those proteins into small fragments here. Maybe biggest machinery in our body is ribosome. Ribosome has about 70S structure, 70S of the size, and proteasome altogether has a 26S. It's smaller than ribosome, but maybe number 2 or number 3, one of those biggest the structure in our bodies. Ubiquitination can be catalyzed by this complex, and by adding dimerizer here, so-called protect, and then target protein can bind here, then this machinery can automatically ubiquitinate over this target protein. So that means you need to design this dimerize. One side, they need to bind to this machinery. The other side is target-specific small molecule. So that means if you have inhibitor or activator, whatever it is, if this small molecule can bind to the target protein strongly, then you can just label this into this side, and then this side recruit this target molecule near to ubiquitination machinery, and then this ubiquitination just can be added. As a result, now proteasome can recognize this target and then destroy those targets. Then ubiquitin can be recycled, and also this machinery is also can be recycled, and it can recruit another target, can make this the catalytic cycle to destroy those proteins and further and further. Quite interesting approach, right? So far, we didn't physically destroy the target protein, but using this approach, we select those targets using small molecule tool, recruit it, just ubiquitinate, and physically destroy the target. Last topic is the gene expression regulations. Previously, we have studied knockout and knock-in. Knockout is deleting one gene from the DNA. Knock-in is introducing new gene into the DNA. To visualize, one method is utilizing GFP, green fluorescent protein. Then the knock-in effect can be easily visualized like this. In comparison to knockout, knockout is complete the deletion of the gene. What was the temporal suppression of those gene expression targeting mRNA? Yeah, we called it as a knockdown, and then knockdown can be induced by RNAi. Do you remember RNAi was discovered from what animal? Yeah, it was C. elegans. So we have a whole sequence of C. elegans, and then RNAi works perfectly in C. elegans. Then what can you do? By reading those genomic sequence, and then we just select only 22 nucleotide long sequences which can match to mRNA of each individual gene. If we can synthesize those double-strand RNA, now we have a very powerful tool which can temporarily suppress the gene expressions. Now this kind of a large scale of screening is possible, genomic scale. Advantage of this approach compared to just a small molecule or the chemicals. Small molecule chemical, we need to find out or discover the small molecule tools which attack one by one for each individual of those proteins. If you utilize this siRNA, now it's possible you just select one gene, and then just by adding this gene in terms of siRNA, and you can knock down, suppress those protein synthesis of that specific gene, then observe what's happening in phenotypic changes. This is really powerful reverse genetic chemical tools. Well, this is kind of the in-between genetics and chemical genetics, but siRNA is synthetically you can prepare. So this is in the boundary of the area. This can be considered as modern chemical genetics approach. Not only C. elegans, seems like siRNA also works in plant and also insect cells. So that means this genomic scale of screening can be expanded to different the species. In mammalian cells, siRNA, we need to use some special transfection reagent, but seems like it's possible to deliver this siRNA to the mammalian cell too. Then, let's say, utilizing drug compounds and screening in high throughput, exactly same format we can do siRNA screening, and even maybe we can do drug and siRNA screening as a combination too. Now this expands our toolboxes amazingly at the wide the view, covering of all genomic, whole genomic at the scale, and also whole natural product and combinatorial library together. We have really powerful tools for studying chemical genetics the work. siRNA has been used as a really powerful technique. Now seems like we are getting another revolutionary technique, so-called CRISPR. I found excellent the introduction movie for this CRISPR, so I will connect you to the movie directly.
Until recently, gene editing was extremely expensive, complicated, and took a long time to do. This has now changed with a revolutionary new technology now entering the stage, CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. It's hard to get across how big a technical revolution CRISPR is. It literally has the potential to change humanity forever. Why did this sudden revolution happen, and how does it work? Bacteria and viruses have been fighting since the dawn of life. So-called bacteriophages or phages hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it's activated and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise, almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell, microorganisms, plants, animals or humans.
How did you feel? Did you feel the real revolution is coming to us? We will see how it goes. This is the end of the lecture of today. And this page tells you that if you have any questions from this lecture, you can put up. And any unclear part also, you can ask me. And also now this is a time you need to seriously think about your proposal. And would you try to put up one topic, almost a semi-final topic, you want to spend some weeks to prepare for your proposal? To remind you, the deadline of your submission of your proposal and also presentation material means 20 minutes of the video clip is October 10. So I think it's the right time you should start. Okay? So again, the deadline is Sunday noon, and see you next week. Bye-bye.